This is Chapter 12, the nervous system and nervous tissue. We're going to be looking at just the basic structure and the basic functions. We'll get into detail in later chapters. So the nervous system can be divided into two parts, if you will. There's the central nervous system, which is composed of the brain and the spinal cord, and then the peripheral nervous system, which is anything outside of the brain and the spinal cord. Now, obviously, there are connections between the central nervous system and the peripheral nervous system because they have to communicate with each other. So those nerves that connect to the brain or to the spinal cord, those would be classified as part of the peripheral. They connect to the brain and spinal cord, but they're not part of it. So that's why they're in the peripheral nervous system. And this diagram is just showing the two different divisions of the nervous system. The basic structures, first you have a broad category of two different types of cells with the nervous system, the glial cells and the neuron. The glial cells are the supporting cells. There are several different types of these. We'll get into in just a moment. But they are the cells that do not transmit electrical charge. They're just supporting the neuron, which is the other type of cell. And the neuron is the functional cell. It can generate this electrical impulse, which is the nerve impulse. The neuron, here we go again, we're going to give a little bit of different terms. The neuron, if you remember from histology, has a different structure to it. The cell body is what we call the soma, and then it has several extensions or processes from that cell body. Typically, on what you would classify as your classic neuron, the axon is going to be the longer process or longer extension. It does extend from that soma or cell body, and it's going to be moving the impulse away from the cell. So it's going to go from the cell body down towards the end of the axon. That's the direction it would be moving. These shorter extensions or shorter processes on the cell body are the dendrites. And these are the ones that are receiving impulses from other neurons. They're going to be moving that impulse towards the cell body. Oftentimes the neurons, you'll hear the description of white matter versus gray matter. This is referring usually to the axons and whether or not they're myelinated. If they are myelinated, they're called white matter because they look whitish in color. If they do not have this myelin sheath around them, they tend to look gray. If you're looking at just a group of cell bodies or a group of somas, that would be gray matter. And so something that you can see here that is kind of interesting, if you will, excuse me, is looking at, over here we have the brain, and if you do a little dissection into it, cut away a piece, this is the surface over here, but with the brain, when you look at it, on the superficial area is the gray matter, and then deeper is the white matter. And one of the reasons I'm pointing this out is when we study both the brain and the spinal cord in detail, you're going to see in the spinal cord it's reversed. The white matter is on the superficial and the gray matter is deep or more interior to it. And this white matter, as I said, this is axons that have the myelin sheaths around them, it's the lipid layer around them, and it gives that whitish color to it. Now, if you were to do a cross-section in the schematic drawing, as you can see here once again, it's easy to depict here that all along the superficial areas is the gray matter, and then internally is the white matter. In this picture right here, I'm just going to point out this area right here. It's called the corpus callosum. It is the connection between the right and the left hemisphere of the cerebrum. So if you look at the basic structure, once again, CNS is central nervous system, and I know this can be confusing, but here we go. When you have a group of cell bodies, it is known as a nucleus. And I know we use this term a lot, a little different interpretations in biology. You can have the nucleus of an atom, you can have the nucleus in the cell. Well, when you're talking about the nervous system and you use the term nucleus, you're talking about a group of cell bodies. I know it gets confusing. I guess we don't have a large vocabulary. I don't know. Track refers to a group of axons in the central nervous system. Now, when we talk about a group of cell bodies or a group of axons in the peripheral nervous system, we're going to use a different term, and this is to distinguish between the CNS and the PNS. PNS, peripheral nervous system. When you have a group of cell bodies here, we call it a ganglion. And when you have a group of axons, we call it a nerve. So we have like spinal nerves, cranial nerves, etc. But it's not talking about an individual nerve, it's talking about a group of axons. So some of the basic functions of the nervous system is sensations that receive stimulus on different changes or different factors in the environment, whether it's cold, whether it's pain, light touch, heavy touch, etc. Then there's the response. This involves motor functions. Some of these are voluntary, some of these are involuntary. As we further explore the nervous system, you're going to find if it is a voluntary response, that's going to involve what we call the somatic nervous system. And if it's involuntary, it's going to involve the autonomic nervous system. And then there's also this step or this process of integration. If you were to put this in order, it would be between the sensations and the response. Basically, what it does is there are some neurons that are going to be interpreting information, this sensory information that arrives. What is this? Now, what do we need to do about it? Do we ignore it, or do we need to have some type of a response? And it would determine what that appropriate response would be. It's cold. You come in a room, ooh, you get the stimulus that is kind of chilly. The information goes to the brain. Okay, it's not too bad. Let's just wait and see. Or, yeah, it's chilly. Put a coat on. Or turn up the thermostat. So as you can see, we talk about the nervous system, but we break it down. By now, you're starting to pick up, hopefully, that biology we left it. Organized stuff. So with the nervous system, first you divide it into the central nervous system versus the peripheral nervous system. Like I said, the central nervous system is just the brain and spinal cord. Everything else is the peripheral nervous system. Now, you can also divide into the peripheral nervous system into the somatic versus autonomic. Somatic, as we just said, is you have voluntary control over it. Autonomic, you do not. It's involuntary. The autonomic nervous system can be divided into subdivisions, if you will. Now, not all books will include the enteric nervous system. This involves nerve responses, excuse me, typically with the abdominal muscles. What is most commonly agreed upon is that the autonomic nervous system, you'll definitely see these two divisions, the sympathetic nervous division and the parasympathetic nervous division. And like I said earlier, we will get into all of these in more detail. Just so you know, sympathetic nervous division, it's involuntary because it's part of the autonomic nervous system. What happens is this is triggered or turned on, if you will, when you are in a crisis situation. When something has happened and your body has detected essentially that your life may be in danger. It's what is commonly referred to as the fight or flight. You're either going to run to get yourself out of the situation, or if you can't run, you're going to stay there and you're going to fight like heck to get yourself to survive the situation. Under normal conditions, the division that's turned on is called the parasympathetic, and this is sometimes called the rest and repose. It's what hopefully you're in right now, where it's kind of, when you eat, when you're just relaxing, you're just chilling out, everything's fine. it is the parasympathetic nervous division that is working or is activated. And so this is just showing that the central nervous system, like I said, the brain and spinal cord, all of these nerves are outside the brain and spinal cord, so they're part of the peripheral nervous system. You have nerves that are picking up sensory information. You have nerves that are sending down the motor responses. And right here, like I said, especially with the digestive tract, is the enteric nervous system. The neuron, as I said earlier, this is the specialized cell, the functional cell, if you will, of the nervous system that is able to generate and move along an electrical impulse. So here is the soma, the cell body here. So this would be the nucleus in the middle. Most of your organelles are going to be in the soma. These smaller extensions are the dendrites. So they are the ones that receive an electrical impulse. So you can see there's in the background here another neuron, and it would have generated an electrical impulse, and it's going to stimulate this one along the dendrites. This longer extension here is the axon. Where the axon attaches to the soma, it's a widening area. It kind of looks like a funnel, and that's known as the hillocks area. Your electrical impulse is going to be traveling away from the soma down the axon towards the end, which is known as your axon terminus. So you can see you have these little branches here. The myelin sheath, as you can see here, it's layers of lipid that's wrapped around the axon. It acts as insulation, and this is made by, and actually is part of, some of your glial cells, your supporting cells. That's what a oligodendrocyte is. It's a glial cells. And especially in the peripheral nervous system, we see what's known as the node of Ranvier. It's the little spaces between the myelin sheaths where there is no insulation. So you can classify neurons either structurally or functionally. If you look at the structural classification of neurons, the three major types, and there are other types, but the three major ones are what we call unipolar. That's where there is just one process that's extending out from the cell body. There is what's known as bipolar, where now you would have two processes extending out from opposite sides of the cell body. The bipolar ones are relatively rare. We do see them in the eye, the type of cells that pick up reception for visual stimuli tend to be bipolar cells. The most common type are multipolar, and that's similar to the picture I just showed you, where you have multiple dendrites and then one major axon. So this is showing unipolar, your cell body or your soma, it's off to the side. Essentially, this is just one process. Bipolar, the cell body's in the middle. Excuse me. So, like I said, you've got these extensions on either side, and then the multipolar, like we just saw previously. So, like I just said, multipolar is going to be the most common. If you look at the functional classification of neurons, now this is where we have the sensory neuron, the motor neuron, and then the integration or association neuron. Integration and association are the same thing. Sensory neuron is receiving the stimulus, and it is going to be sending that impulse towards the central nervous system. The motor neuron is receiving an impulse from the central nervous system, so it's moving away from the brain and the spinal cord. The integration or association neuron, usually that's embedded in the central nervous system. It's receiving the impulse from the sensory, and then it's sending an impulse to the motor neuron. So they're like the middleman. For glial cells, these are the supporting cells. In the central nervous system, there are 4 different types. In the peripheral nervous system, there are only 2. And of course, you know, to make your life easier, we're going to give them different names so that we can, when you say the name, you know exactly which location we're talking about. So in the central nervous system, we have the astrocytes, the microglial, the oligodendrocytes, and the epidermal cells. And then in the peripheral nervous system, we have the Schwann cells and the satellite cells. So in the central nervous system, your astrocytes, these tend to be star-shaped. I'll have a picture in a moment, and yeah, they look like a star. They're very much involved with helping to support the neurons. They are definitely involved with what is known as the blood-brain barrier. The brain is very sensitive tissue, and obviously of major importance. It's controlling everything. So you want to be very careful of any chemicals that may be exposed to the brain. In other words, everything that you take in your body, you don't necessarily want it to come in direct contact with the brain. So as a protective mechanism, what has happened is that at the cellular level, things have developed in such a way that you have these astrocytes, that anything that is in the blood, if it starts to come out of the blood, diffuse out, before it can touch the brain, it has to pass through the astrocytes. So it's sort of like a filter, and some things cannot pass through it. The oligodendrocytes we saw just a little bit ago, that helps provide the insulation. And think of it, that makes sense. If you're talking about electrical charges moving down the axon, I mean, if you think about electricity, the wires in your house, okay, I'm not an electrician, but even I know that you want insulation around those wires. You want to help protect it. Well, the oligodendrocytes are essentially like your insulation around the wires. Microglial cells, these are specialized cells that do carry out phagocytosis, so they engulf anything that is deemed or recognized as a foreign substance. So obviously it is a protective mechanism. The epidermal cells, they also help to filter blood, and they are involved. Later as we study, we will see some of these are specialized in a certain area, excuse me, to make cerebrospinal fluid. So in this picture, we can see up here, this yellowish color, that's the neuron. And down, follow around, you've got these different axons, because you got several different neurons here. So here's a neuron, here's a neuron, there's another one up here. These long yellowish strands are the axons, and this, I don't even know what color to call this, turquoise color, this would be the oligodendrocytes. They're very large. So you have the main cell here, and then you've got these extensions off of them, and they wrap around that axon, providing that insulation. So one oligodendrocyte actually can have contact with several different neurons in terms of on the axons, and it also kind of helps bridge them together. So I'm sorry, I'm trying to do this with my cat, who has decided she wants to either have me stop or wants to participate with this. So I apologize for background noise. It's my cat. So here are the oligodendrocytes. See these star-shaped cells up here? Those are the astrocytes. And notice how, yes, see, they're helping to support. Now, it's not showing the blood vessels here, but anything would have to pass through the astrocyte before it gets to the neuron. Here's a microglial cell up here. There's another one right down here. As I said, those are going to be able to move around freely through all the spaces here, and they can detect what's normal. They know this neuron is a normal cell. It's part of you. It can recognize what we call self versus non-self, so it leaves you alone. But if there was a bacteria here, it would attack and destroy it. And then down here, your epidermal cells, as I said, we're going to see where those are going to be producing the cerebrospinal fluid. In the peripheral nervous system, remember you only have two glial cells, or two types of them. The satellite cells, they're helping to support. And then the Schwann cells, they're providing the insulation. So they're similar to the oligodendrocytes just in the peripheral nervous system. So here are your satellite cells around a neuron and then a Schwann cell. Now the Schwann cells, one difference between them and oligodendrocytes, notice they don't have extensions off of them reaching to another neuron. They just wrap around, completely wrap around the axon. So what is this myelin sheath, this wrapping around of these cells? It's mostly composed of lipids. So it does provide the sheath or this coat, and like I said, it provides insulation. It does have a whitish color to it because of the lipid composition in it. The electrical impulses we have found, we're able to measure it, which I don't know. Okay, I'm a science nerd, but I find it fascinating. We can actually measure it on an individual cell. And what we have found is that the electrical impulse does travel much faster down a myelinated axon than a bare axon. That insulation helps to keep that charge from spreading out and dissipating. So it keeps it just going straight down that axon. It's going to move much faster. So here is the axon, and you can see from the cell, the Schwann cell, how it starts to wrap around until it's very tightly wrapped around here. And then here is really, once again, okay, I'm a science nerd. I find it fascinating. Look at the picture. So here is the neuron itself, with the axon extension. You did a cross section. Here's that myelin sheath around here. So a stimulus, whatever that stimulus may be, it may be pain, it may be light touch, it could be hot temperature, cold temperature, whatever, there's a stimulus. And it's going to produce a graded potential. Graded potential is just a localized change in the electrical charge in the cell. If that graded potential, that change in the electrical charge, reaches what we call threshold, then an action potential is generated. So you have to have a graded potential first. You have to have just this little localized change in the charge of the cell. Like I say, if it reaches a certain point, which is the threshold, then you will generate an action potential. If you don't reach threshold, you'll never get the action potential. Propagation is a term that we use to describe the action potential that's traveling down the axon. When the action potential reaches the axon terminus, the very end of the axon, a neurotransmitter is going to be released. We're going to go through all this. So you have, as it says here, a lot of sensory endings, especially in the hand, you've got a lot. So you pick up a stimulus. And what happens to that stimulus? Okay, it triggers an action potential in this neuron. This would be your sensory neuron, and that action potential is traveling down the arm from the hand. It goes up the arm, to the shoulder, around. Now here we go. Excuse me, this is the sensory neuron. Boom, it's in the spinal cord. Then it's going to go up the spinal cord. Here's the end of the sensory neuron. It's going to connect with an inner neuron. Ooh, okay. We're getting a stimulus. Now it's showing here, so you're in, let's say, the shower, and let's say it's really hot water. And so the inner neuron's picking up that information. Let's interpret it. Okay, we're going up here. We have to now bring it to the appropriate portion in the brain, interpret that information. What do? Oh, you know what? It's really, really hot. Maybe we need to move the hand. Okay, send that information now down the motor neuron. It moves down through the brain, down the spinal cord, out here. through the shoulder, down the upper arm, down the lower arm, down the wrist, down to the hand, which is going to say, move your hand, get it out of the hot water. And obviously, all of this, fractions of a second. So how do you generate this graded potential and the action potential? We're talking about a charge in the cell, and we have to change that charge. How are you going to do that? It's by changing the ion concentration. Things like sodium, potassium, calcium. Actually, all of those have a positive charge to them. Chlorine has a negative charge to it. You're going to be changing the concentrations of these, and that's going to change the charge inside the cell versus outside the cell. And in order to understand how do you change this? Well, keep in mind the cell membrane. So this is the cell membrane here. Remember, it is that phospholipid bilayer. In the middle here, it is hydrophobic. So a lot of things cannot pass directly through the membrane. Things typically that have a charge to them, ions, typically are hydrophilic, meaning they will mix with water, but they don't like the fats here. And so typically they can't just pass through easily. They will need help of some of these proteins that are transmembrane proteins, just meaning they extend the width of the membrane. So they're going to act as a transport system. A lot of them will have these pores or openings in here that will be usually very specific in what ion they will allow to pass through. So that cell membrane, remember, is hydrophobic in the middle. Charged particles need help to get across. And something that is very, very critical, it has been studied extensively, we know exactly how it works, is the sodium-potassium pump. What the sodium-potassium pump does is you naturally have some sodium that is leaking in and potassium is leaking out because it's trying to reach equilibrium. You don't want that. You don't want it to reach equilibrium. So the sodium-potassium pump is going to carry sodium out of the cell, and it's going to bring potassium in. And in this way, it can help to regulate the concentration of those two ions on both sides of the membrane. If you are a very visual person, there are a lot of things on YouTube where you can see where people have made little animated short movies of showing the sodium-potassium pump. Some of them are really good, some not so good. So you might just have to search a bit. But if you're visual, it can be very helpful in looking at that. There are different types of transport proteins. There are different types of what we call channels. As to how, basically it relates to how do you open and close them. Excuse me. So a ligand-gated channel, a ligand is just a chemical. So in studying how muscle contractions work, you've already seen an example of this. This is where a chemical has to bind to the protein in order to open it up. It's kind of like you got to stick a key in here to open the door. What compound is going, or what chemical is going to open this, it has to bind to these little receptor sites. And they are very, very specific. So that's why I say it's kind of like in order to open your car door, you have to have your key. My key, car key, is not going to work on your car, or at least it shouldn't. We hope not. It's very, very specific. So in this case, what is the chemical they're talking about? Oftentimes they're talking about neurotransmitters. Those are chemicals, so they would be a ligand. The one that you're familiar with already is the acetylcholine. If it's not acetylcholine, it's not going to bind here. It's just going to continue on its little way. If it is acetylcholine, as you can see here, it is going to bind. When it binds, what does it do? How many times have you heard me say, whenever you have two things bind, you're always, always, always going to see a shape change. It may not be real obvious, but there's going to be a shape change. And here you bind In this case, it's showing the acetylcholine, but you bind a neurotransmitter, which is a ligand. A ligand is just another molecule. When you bind it, when you put the key in the keyhole, it opens the door. And now look out here. You've got calcium out here. You've got sodium out here. You have potassium inside. Well, guess what happens? You have a lot of sodium and calcium outside the cell. Very little inside. You don't see a whole lot of purple in here. You don't see any green in here. So when you open, think of it, when you open the door, these allow sodium and calcium to move inside. And it's going to allow, depending on the gate, sometimes, like I say, some of these are specific. You would allow then the potassium to move out. They're flowing down their concentration gradient. But a ligand-gated channel just means that you have a chemical that's going to bind, and that's what triggers the opening of that channel. You remove that chemical, and then it's going to close. Closes the door, if you will. And the most well-studied one is the acetylcholine. Now, some of the gates can be mechanically gated channels. So you have here, it's closed. Notice the difference right here with the cell membrane. This often happens with things like some of your pressure receptors, is how they become activated. There's pressure placed on. Essentially, the sides are squeezing in, and that's going to pop open the gate, if you will. So it's opening due to some type of an actual physical distortion or disruption of that cell membrane, like pushing in. Oh, it's squeezing it in, and it's going to pop the lid off, open the door. You restore it back to its original shape, and the door closes. Voltage-gated channels. We see a lot of these, and you've already been exposed to this a little bit with muscle contraction. Once again, you still have your transmembrane protein here. Here's your cell membrane. What happens is at rest, it's closed. You have a change in the charge. Look right here. At rest, a cell typically is, we can measure the voltage, and it's around minus 70 millivolts. If it changes to a certain point, it's not going to minus 69. It has to be a significant change. So right here, it reaches minus 50, boom, it opens. That change in the charge opens the gate, and now it allows ions to pass through. If you restore and bring the charge back to minus 70, it's going to close again. So you've got your ligand that responds to chemicals. You have your mechanical that responds to distortions. You have your voltage gates that respond to electrical charges. And then you have some that are referred to as leakage channels. And some of these just essentially are open all the time. And some of them are just random as to when they open or when they close. And it allows ions to move. When any of those gates open, regardless of what kind it is, whether it's voltage, whether it's ligand, leaking ones, etc., things are naturally going to want to move from an area of high concentration to an area of low concentration. That's what diffusion states. The resting membrane potential. So when a cell is at rest, like I said, we can measure the voltage. It's around minus 70 millivolts. At a resting cell, the sodium level or concentration is much higher outside the cell. Sodium has a plus charge to it, +1 charge. Potassium, which also has a +1 charge, is higher inside the cell. Usually in the cytoplasm, in the cytosol, the liquid portion of the cell, usually calcium is very low. It's going to be higher outside the cell, because you have some chlorine and other ions inside. Basically what happens is outside the cell is positively charged, inside the cell is negatively charged. How do you maintain that? Well, you have got some of these leakage channels. So like potassium, we know there are leakage channels with potassium. Potassium is wanting constantly to flow out. It's higher inside, so it wants to flow outside. How do you maintain this minus 70 millivolts? How come it doesn't, if it's negative, how come it's not going more positive if the potassium is constantly leaking out? That's where you have to have ion pumps like the sodium potassium pump to restore and keep the balance, which is going to store and maintain that negative charge on the inside. You have to keep the cell negatively charged inside, because if it were to ever reach equilibrium, so there would be no charge, then whenever you were to receive an electrical impulse, it's not going to cause any changes, because you don't want equilibrium between inside the cell and outside the cell. Ultimately, you would never get electrical impulses, you would never get muscle contractions, and you would not survive. So with an action potential, you'll see that we often talk about depolarization, repolarization, and hyperpolarization. So what are we talking about? If the resting potential or the resting membrane is at minus 70 millivolts, depolarization is when you change that internal charge to move it more positive. You're moving it towards zero. If it's at minus 70, you're going to move it towards zero because you're trying to go to more positive. How is this happening? Because you have an influx of sodium. Sodium is pouring in. Sodium has a positive charge. So if the inside's negative and you have all this positive charge coming in, you're going to see a shift, and it's going to be shifting towards more positive. That's depolarization. Repolarization is when you're trying to move the potential back towards minus 70 millivolts, which is the resting state. So you're trying to bring it a little bit more negative there. You're trying to restore it back. You would have had depolarization first, so you brought it more positive. Now you're trying to get it back to that resting state. How are you doing that? How are you trying to get it more negative again? Because you have the efflux of potassium. The potassium is flowing out of the cell. Potassium has a +1 charge. So internally, inside the cell, it became more positive. Now you're moving that positive charge out. And you've closed your sodium channel, so you're not letting any more sodium come in. No, don't let any more positive sodium in. You're trying to get it more negative. That's repolarization. Hyperpolarization is when you're moving that membrane potential below or more negative than the resting state. So you're trying to inhibit anything from happening. So you're going actually more negative than minus 70. And we can graph this. So over here, if we start on the left-hand side, this is at resting. And then typically what happens is you open some of your sodium gates. If you're a timeout in a muscle, what's happening? Typically, first you're opening a ligand gate. So in the case of muscles, you had acetylcholine bind. That triggered the opening of the sodium gate. So the sodium is coming in, the plus charge, it's making inside the cell. You're getting more positive now. You get more and more positive. That charge triggers the voltage sodium gates to open. So now you have an even more rapid increase or influx of sodium. This is all depolarization. You're becoming more positive. Now you're actually at a +30 charge. This dotted line right here is threshold. That's kind of the magic number. If you only got to here and you didn't quite reach that -55, nothing would happen. Once you hit threshold, it's all or nothing. Boom, we're gone. We've got the action potential. Now your sodium gates are going to close. Your potassium gates open. Potassium is high inside the cell. So when you open those gates, that positive charge is now flowing out. And you're not letting any more sodium in. So inside, you're starting to become a little bit more negative. This is repolarization occurring. You're trying to restore back to resting. And like anything, it's hard to exactly hit it, so sometimes you overshoot it. So you're actually below -70. This area is hyperpolarization. And then finally you restore back to resting. So if we look at the steps of the action potential, the ligand-gated channel, something bound to it. Like I said, muscles, it's going to be the neurotransmitter acetylcholine. It may be a different neurotransmitter. If you have a nerve to a nerve, it's going to be a different neurotransmitter. The ligand-gated channel, though, it's going to open, and this is very specific for sodium. It allows sodium to come pouring in. Influx of sodium just means you've got sodium flowing into the cell. Sodium has a plus charge, so the internal part of the cell is now becoming more positive. That is depolarization. You're generating depolarized cell here. As you have that influx of the positive charge, as you can see, it's going to cause the voltage gate now to open because you're changing the charge. Now you're going to have an additional influx of sodium resulting in even greater depolarization where you have that high peak. You reach that threshold, the action potential spreads now through the whole length of the cell membrane. When the charge is just localized, we refer to it as a graded potential. But once you reach that threshold and that charge goes down the entire length of the cell membrane, that's an action potential. So think graded is just local. Action potential is going to go the entire length of the cell. When the sodium gates close and the potassium gates open, that's going to cause the repolarization. How long does this whole process take? About 2 milliseconds. So not very long. The refractory period is a time period that when the action potential is in progress, another one cannot be started. You have to restore everything back to the resting state before you can generate another action potential. So if you haven't restored everything yet, nothing else can be started. There's two phases. There's the absolute refractory period where another action potential is not going to start. Now there's an area that we call the relative refractory period where an action potential, it could only be started if the stimulus was stronger than the initial stimulus. Graded potential, I said that's just a local change in that resting membrane potential, that local charge, not the entire length of the cell, just in a small area. The postsynaptic potential, this is the graded potential in the dendrites of the neuron, the one that's receiving this impulse from another cell. So we talk about presynaptic and postsynaptic. Presynaptic is the one that would be generated, sending the impulse. Postsynaptic is receiving. Think of it almost like if I were to extend my hand to shake your hand, I would be the presynaptic one. I'm extending my hand to you. You would reach out to take it and receive it. You would be postsynaptic. Now there's two types of postsynaptic potentials. There's excitatory or inhibitory. Excitatory is going to cause a depolarization, get you closer to having that action potential. Inhibitory is what we call, it's going to cause hyperpolarization. It's going to, as the name implies, try to inhibit the production of a graded potential, make it even further away from that threshold. There are certain chemicals and drugs that are called excitatory, and some that are inhibitory. Caffeine often initially can cause excitatory, where it brings you closer to that threshold, which is why some people seem more excitable when they're taking caffeine. Synapses, the chemical synapse, that's a neurotransmitter that's released from the neuron, and it's going to affect whatever that postsynaptic cell is. It could be another neuron, it could be a muscle, it could be a gland that, say, secretes a hormone or something. But a chemical synapse is where the neurotransmitter is being released. Electrical synapse, this is where there's direct connection between two cells. So the ions are going to pass directly from cell to cell to trigger that depolarization, all kind of as a unified step. What we see mostly are the chemical synapse. So what are characteristics of a chemical synapse? There's going to be a presynaptic element, typically a neuron. There's going to be in that presynaptic element a neurotransmitter that is packaged in vesicles. There is a synaptic cleft. This is the space between the presynaptic and the postsynaptic element. On the postsynaptic element, which has to be there, which can be, like I said, another neuron, it could be a muscle, it could be a gland, and on that postsynaptic element will be receptor proteins that are very specific for binding that neurotransmitter. And then that neurotransmitter, that's great for starting up the whole process, but how do you stop it? There needs to be some way of, if you will, deactivating that neurotransmitter. There has to be some method of either breaking it down and eliminating it or reabsorbing, taking it back up again. So in this diagram, it is showing here your presynaptic neuron. You have these little circles. So these are all vesicles. They are containing your neurotransmitter. So when an axial potential comes down, as we're going to see, what's going to happen is these right here are voltage gates, typically for calcium. So when the charge comes down along the axon, it's going to trigger the opening of these calcium gates. Calcium, which typically is higher outside, will flow in. That is going to trigger these vesicles to move towards the membrane here, and then by exocytosis, release the neurotransmitter. This is the synaptic cleft. This is that space between, in this drawing, you've got a neuron over on the side. So it'd be between the presynaptic neuron and then, in this case, the postsynaptic neuron. When the neurotransmitter is released from the presynaptic neuron, it's going to diffuse across the synaptic cleft. Now over here, these are receptors that are very specific for this neurotransmitter. They're going to be a ligand-gated receptors, so the neurotransmitter binds to it, and that's going to open, and now you're going to start the whole process here on the postsynaptic neuron. So that neurotransmitter release, like I was just saying, the action potential reaches the axon terminus. It triggers the opening of voltage gates that are specific to calcium. So it opens those calcium voltage gates, resulting in this huge influx of calcium into that presynaptic neuron. The calcium binds to those vesicles that contain the neurotransmitters and causes the vesicle to merge with that cell membrane, releasing the neurotransmitter. The neurotransmitter is then going to diffuse across that synaptic cleft and bind to the receptor protein of the postsynaptic cell. And now you're going to trigger the whole effect on the postsynaptic cell.